So today we are going to talk about the structure of RNA and what it has in common with DNA and what is unique to RNA that's different from DNA. And I'm hoping to keep just the structure of RNA video pretty short. So let's jump in. I'm starting out in our unit packet on page 17. So if you want to follow along, you're welcome to read all of this if you want to. So I'm going to go ahead and just jump down to the blanks and help us get started. Okay, so a nucleotide. And if you go back, you'll remember that in our very first unit of the year, we got into organic compounds and macromolecules, and we talked about carbohydrates and lipids and proteins and nucleic acids as the big four. So, what is a nucleic acid? It is something that is made up of a nucleotide, which is like the little tiny building blocks, the little baby Legos that make a big Lego castle. So, for DNA, the Na stands for nucleic acid. So, let's talk about it. So, to be a nucleotide, you only have to have three things, which is great. You have to have a phosphate group, which is usually represented by a circle. So let me see if I can show you here. So this circle right there, all of those are going to be phosphate groups. And then the next thing that is required to be a nucleotide is to be attached to a 5-carbon sugar. So those are usually represented by pentagon or little house shapes. And if it's DNA, that sugar will be deoxyribose, thus the letter D for DNA. And then if it's RNA, that sugar will be just ribose. And that's why RNA is starting with an R for ribose, right? Ribonucleic acid. And DNA starts with a D for deoxyribonucleic acid because they are named after the sugars ribose and deoxyribose. So that's how you get the name for DNA and RNA. The last component of a nucleotide would be a nitrogen base. So sometimes you hear those called nitrogen bases, nitrogenous bases, or nitrogen containing bases. It's all the same, okay? And even though they have some different shapes, all that I personally need you to know at this level is that those are nitrogen bases. So in DNA, there are 4 nitrogen bases that you can see in the picture on our screen. So you have adenine, guanine, cytosine, and thymine. And those are the 4 DNA nitrogen bases. If we are in RNA, there is only one difference. You still have adenine and guanine and cytosine, but instead of thymine, you have something called uracil. And it basically is just a substitute player. It's a substitute nitrogen base that RNA uses instead of thymine. All the bonding patterns, when you pair up nitrogen bases, still apply. Everything is still as it normally is. You just substitute a U instead of a T. And that's the only difference in the structures of their nucleotides. So I'm going to bounce back up here. Let's see here. Uh-oh. Here we are. Okay, so in terms of the blanks that are present on your worksheet, what makes up a nucleotide? Well, what makes up a nucleotide is first, phosphate. Oh, phosphate. There we go. The second thing that makes up a nucleotide would be a 5-carbon sugar. That's the easiest way to, instead of saying deoxyribose or ribose, we just call them 5-carbon sugars. That way we don't have to specify until we need to. The last thing you need would be nitrogen bases. And those would be our adenine, cytosine, guanine, thymine, or uracil. Okay, blank, 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 and blank are 4 kinds of bases in DNA. So rather than write out all of those things that I just mentioned, like adenine and cytosine, I'm just going to abbreviate. So A for adenine. Okay, and then over here, I'm going to put T for thymine. and C for cytosine, and lastly, G for guanine. And then it asks you which one is unique to RNA. And I'm going to go ahead and write out uracil as a whole word just so that you guys have a copy of it somewhere. You're welcome to abbreviate it as just a U if you want to. All right, what shape did Watson and Crick determine the DNA model is? So there's a couple of choices that you could write here. Some people say twisted ladder. I prefer to call it a double helix. Oops. And that's probably a term that a lot of you have heard before. Basically, the only reason people call it a twisted ladder is because DNA looks like a ladder. It's got the parts on the side where you would put your hands and then the rungs in the middle where you would climb up. And if you take a ladder and you turn it and twist it, it looks just like a double helix. So that's why people call it a twisted ladder. Okay, so let's answer this fourth one. Name two differences in DNA and RNA. So one difference is that DNA is double-stranded. That's why it's a double helix. And RNA is single-stranded. And so it is a single helix. I like to refer to RNA visually as like a curly fry or like one little spiral of hair, something like that. Okay, another difference between DNA and RNA could be the type of sugar. You could have put deoxyribose instead of ribose, or you could pick which nitrogen bases. So you could put DNA has thymine, while RNA has uracil. Really, you could put their functions, which we'll learn about in another video. Anything that you notice that is a difference is welcome to go in that blank. As far as what is the job of DNA, it is to pass hereditary information from parents to offspring. Let me mark that down. So the whole goal of DNA is to simply hold your genetic information and try to pass it in case you ever want to procreate and have a child. And it's about the survival of the human species as a whole. It's not so much us as individuals as it is our whole species being able to survive. And so it's just a mechanism or a way or a pathway for information genetically to go from point A to point B for the survival of the species. Okay, so we will continue practicing and learning more in another video. I hope this was helpful. Thank you so much.